today I'm going to talk about protein nanoparticle engineering for vaccine development, and really just to give a little bit of background on the shift in vaccines from the typical vaccines that most of you have gotten and are still getting from live inactivated or killed viruses or pathogens towards subunit vaccines. And so some of the difficulties with whole pathogens are you've got everything is there. So all sorts of different surface proteins, lots of different components, and so you have an immune response that is broadly responding to a lot of different antigens. And so while that can give you, make the pathogen very immunogenic and drive a response, you can't necessarily control exactly what kind of response you're getting. And of course there is always the danger of reversion in case of pathogens that are not actually killed. And so sometimes those off-target responses are very obvious in something like a cancer vaccine, but even in infectious disease, an off-target response can kind of focus the body's immune response towards something that is not productive to actually fighting disease. And so we want to be able to focus the immune response onto the targets that will best protect the patient. And so you can kind of zoom in and instead of vaccinating with an entire pathogen, you can, for example, vaccinate with a single protein or a combination of proteins from the surface of that antigen or of the pathogen. And so this is safer. This cannot replicate. It's protein. The immunogenicity starts to go down as you remove a lot of those components from the pathogen. And the benefits of narrowing your target are then offset by the lack of immunogenicity. You also can have sometimes difficulty creating these proteins. As you know, proteins have to have a folded structure often in order to be recognized and mount the proper immune recognition and response. And so creating recombinantly large proteins and presenting them in a folded function can be difficult. So sometimes we then can zoom even further. And so instead of taking an entire surface protein, can you simply take particular epitopes from that protein or groups of epitopes? And so now we know that we have exactly the target that we want. We can sometimes synthesize them synthetically instead of recombinantly, but both can be done. But the problem is getting worse in terms of immunogenicity. You've now even made a smaller protein, and so the immune response can be basically non-existent in some cases to these kinds of proteins. And as well, the structure itself is smaller, and so you can have things diffusing away from the site and basically getting cleared from the body very quickly. So despite those cons, the benefits of being able to correctly target the immune response toward the proteins that you want is really a benefit. And so a lot of work is moving in that way, and that's the kind of focus that I want to present here. And so then the challenge becomes, well, how do we make these more immunogenic? If we want to give a small peptide or a small protein subunit, what can we do in order to increase the efficacy of these vaccines? And so the typical thing that's done, right, is adjuvants are provided. And so these are inducing innate immune responses. There's well, not a lot of them, but several of them that have been used for a long time, and you have all received them in your whole pathogen vaccines. And the drawbacks there are that you can have overstimulation. There can be side effects. Also, there's a real lack of understanding as to what is actually going on with a lot of adjuvants. Another approach is to try to use nanoparticles or some sort of self-assembling material. And so by inducing formation into a nanoparticle using polymers or lipids or inorganic materials or even other proteins, you create a larger physical structure. So diffusion away from the injection site is slower, so you can keep everything concentrated. The problem is that you've now introduced another kind of material. So while you got rid of the pathogen and all the materials that you didn't want from it, you've now introduced a different kind of material. And so you have the same problem there, right, that you can induce off-target immune responses. Now maybe it's to a polymer or a lipid instead of to some bacterial or viral components. And so while this may have benefit, you're still kind of fighting the same sort of battle. And so the materials that we have been making in my lab are called protein or peptide nanoclusters. And the idea is still to keep the benefits of making a nanostructure and only have the components that you really want, only the subunit antigens that you want to give an immune response to. There's a lot of seats up here if you guys want to come over here. And the idea is we don't want any carrier materials. And so there's no blue in this picture because the idea is, can we literally create our nanoparticle only from the protein or peptide antigen with no other materials at all. And so you're not going to have an off-target immune response, but you still gain the benefits of this kind of nanoparticle, larger object that can stay around and have some other immune benefits. And so that's what the system that I want to talk about today. First, I'll kind of go over generally how we make these and some of the benefits that we've seen in a number of systems, because we've made these from a lot of different antigens. And then in the second half of the talk, I really want to focus on flu and the progress that we've made in developing vaccines using this method, using different flu antigens with the ultimate goal of producing a broadly protecting vaccine. And so the way that we make these nanoclusters, so peptide or protein nanocluster, or PNC, that's what you're going to see on the slides, is that we have in a beaker or in a little vial or even in a big container a solution of our peptide or protein antigen. It usually is just in saline. And then we drip in what's known as a desolvent. And so the desolvent often is ethanol, it could be methanol, acetone. There's a number of different ones that you can use. And as we kind of drip this in, we are inducing nanoprecipitation of the proteins. And when we do this, we form now nanoclusters. The issue is if you take the desolvent away, your proteins are all going to resolubilize again, and so you won't have a stable particle. And so what we need to do then is actually create some covalent linkages. And so what we do is we introduce molecular crosslinkers, and so often these are amine reactive crosslinkers that will simply kind of glue the outer proteins in this particle together, and the crosslinker can be varied. We have used ones that are what I would call kind of permanent. They don't have any sort of reversible bonds in them, there it is. Or you can have a crosslinker with some sort of group in the middle, like a disulfide bond here, where the disulfide bond is sensitive to the reducing conditions in the cell, but it is stable outside of cells where the conditions are more oxidative. And so you can have a stable nanoparticle outside and then in theory a less stable particle inside. And so just in terms of the making and the characterization of these, what I'm showing on the right is kind of an example distribution using dynamic light scattering, where you can see that the diameter of this particular formulation is approaching 200 nanometers. It has a low polydispersity index. Below 0.2 is considered to be good for nanoparticles. And the other thing to see over day 0, 1, 5 is that the distribution is not changing. We're not getting aggregation of these or changes in the structure. And so what I don't want to go over today, but just want to mention to you, is that there's a lot of factors that change the particle properties. And so some of them we can control and some of them we can't. So the first couple are the hardest to control, protein or peptide properties, right? Well, those are dictated by your disease and your antigen. You don't get to control the amino acid sequence. Sometimes you can control the size if you make fragments or fusions, but the protein properties actually significantly affect the particles. Also the protein concentration, right? Sometimes you have solubility limits and there's only so much you can do. In general, higher protein concentration gives you smaller particles. So the things that we can control better are our choice of the solvent and the desolvent, the ratio and volume of each, the volume of each and the ratio between them, and what kind of crosslinker we use, how much we crosslink. And then stirring and temperature are a little bit important, more in some cases than others, but ultimately don't have a big effect. And so the motivation for nanoparticles in general, and then what I'm going to show you are some of the benefits that peptide or protein nanoclusters can improve immunogenicity. And so there was kind of initially this idea for nanoparticles in the literature that you create this depot effect. So if you have nanoparticles, they're going to hang around longer. They're going to be providing antigen over a longer period of time, and so that would give you a benefit just from a kind of a delivery point of view. But it's also been realized along the years that nanoparticle antigens actually have adjuvancy effects in their own right. And so as you present an antigen as a nanoparticle instead of soluble, you get benefits from this kind of repeated multimeric display that can be recognized as more foreign and more like a pathogen looking. You have more uptake of the antigen by antigen-presenting cells than you do in the soluble case. And because it's particulate in nature, the act of endocytosing or phagocytosing these nanoparticles actually stimulates maturation, presentation, processing, and production of cytokines and other signaling molecules by the antigen-presenting cells. And so these are things that kind of are this, what I would call an adjuvant effect that nanoparticles in general, and we see with our protein nanoparticles as well. The function, though, is certainly a factor of what the antigen is that you're delivering, as well as the size, the delivery route, and the spatial organization, where you put the antigens in each kind of particle. Okay, so to just go really quickly, some generic benefits that we see. Some of the benefits that we see are increased uptake, and so what you can maybe see over here on the far left is we have, this is the fluorescence of plain dendritic cells have taken up nothing. This red peak here is the soluble, and then all of these other overlying curves are small, medium, large nanoparticles containing ovalbumin, so a model antigen. And so we get more uptake. Another thing that you see in the middle is that once these particles are getting taken up, the normal route for an endosome to lysosome is acidification and ultimately destruction, which is not useful if you want to be able to prevent your degradation of your antigen completely. And so what we can see here is that the soluble curve is the bottom purple one. It has the most acidification, where all the nanoparticle groups are much higher up. So we're getting lower acidification of nanoparticles regardless of their size. And finally, as an example of an important cytokine in immune responses, IL-1 beta, what you can see here is that some of the formulations, particularly small and medium particles, are having significantly high levels of IL-1 beta relative to our soluble formulation or none at all. And so you can start to tune the properties of your particle to try to gain some of these benefits. Furthermore, we see increased peptide presentation on dendritic cell surfaces after taking up these materials. We see increased MHC1 presentation and 2, depending on what sort of antigen we're presenting, and also increased maturation markers. So I'm not going to point to stuff because it's really hard to get the pointer out there. But you can, the bars are there and the statistics are there. And so another kind of delivery benefit is increased retention at the delivery site. So what you can see in the pictures on the right-hand side, and then it's quantified on the bar graph on the left, is that the nanoclusters at the intradermal injection will stay around for several days, whereas the soluble protein is going away basically immediately. And so we are getting this kind of bolus, or not bolus, but a sustained depot type effect. Okay, so getting to influenza, we really have a need for a broad protecting vaccine. And the seasonal flu is one thing, and you know that some years the flu vaccine works well. They give you the right strains, and it protects us well, and other years it does not protect us very well because of this high variability, and ultimately we never know what strain of flu we are going to be exposed to as it is mutating rapidly. Also, when we have pandemics pop up, people are impacted immediately, and you know how long it takes to get a vaccine, researched, developed, manufactured, given to the population, that our people are dying, immediately from pandemics, and there's not enough time to make new vaccines in response to something that ultimately may not even be around by the time you make the vaccine, because the mutations are so quick. And so I want to go over a few of the important proteins in influenza and basically try to highlight which ones are important and how we're using them in our subunit vaccines. And so when you look at influenza, generally you have hemagglutinin is one of the main surface proteins in purple. You have neuraminidase, these spikes. There are also the red little dots are M2 ion channels. And then there's a couple other proteins that are not pictured here. And basically the HA and NA, so hemagglutinin and neuraminidase, are the most immunogenic. So when you get a whole flu vaccine or you get the flu, you're usually developing antibodies and recognition of the HA and NA. And the problem is those are the proteins that mutate the most. And so while you get that vaccine one year, your response is to those very specific surface epitopes on the edge of those proteins. There's a lot of seats over here. And that is not going to protect you the next year when you get exposed to a mutated virus. And so those proteins, that's not to say that they shouldn't be used at all, because as you can see in the picture here, there's kind of a head and a stalk. And so the stalk region of HA and NA are actually much more conserved. And so the stalk region of HA is one that we're going to be using. The M2e ion channel, the red one, that also is a highly conserved protein. So it is, changes very little across many strains. Unfortunately, it's very poorly immunogenic. So while it might be a good antigen target, the immune response to that is very weak. And finally, nucleoprotein is also a very conserved protein in flu. It's actually on the inside, so it's not something that is often seen on the surface of the flu, but it is widely conserved. And when you are getting normal influenza vaccines, you don't have any response to the NP protein at all that you're developing. You're really concentrated on HA and NA. Okay, so I just want to walk through a couple different kind of generations, if you will, of some of the protein nanoclusters that we have made using these different flu vaccines, so I can show you kind of what we learned and how we've made them fancier, although not more complicated to make over the years. And so the first one we started with was this M2e protein. Because it's highly conserved, we asked the question, could we take this non-immunogenic protein and use our peptide nanocluster idea to group a bunch of them together and actually make it immunogenic? And so the first thing that has to be done is once you start taking small components of these proteins, we need to make sure we're preserving the quaternary structure of these. So M2e is supposed to exist as a tetramer, and so we first had to engineer the ectodomain, so the outside domain of M2e, and we cloned it as a fusion protein with a leucine zipper domain. The name's not important, but basically it gets it to self-assemble into tetramers. So you have more of the natural looking structure of the protein, even though it's not at all on its normal place on the virus. And then we prepared nanoclusters using the method I showed you, using glutaraldehyde, which is a non-reversible crosslinker, as our very first try. And what I just want to show you here is the M2e sequence that we use is on the top. And you can get an idea from different subtypes of flu that there are very few changes noted by the red amino acids as you go from some subtypes to another. So to keep in mind, Puerto Rico is very similar to our nanoclusters. Philippines is identical, while Vietnam and California are more divergent. And so when we made nanoclusters and immunized mice using actually intranasal delivery, we measured antibody titers as well as cellular responses. And I don't want to walk through all of the data and all of the groups in detail because it's a little bit hard to see. But group 1 is the soluble protein alone, while group 2 is the nanocluster, and group 3 is the nanocluster containing CpG, a nucleic acid adjuvant, thinking that if you add another adjuvant, you might get a beneficial response. And actually what we were surprised to see is that group 3 never performed any better than group 2, that we, in all measures, whether we were looking at antibody responses to the M2e peptide itself, so to see if we get M2e recognition, or to see if we can get antibodies that bind the different flus from the different strains of flu. In all cases, you can see the G2 and G3 groups are high, indicating that we are getting good antibody responses to either the M2e peptide alone in cell culture or to the whole virus. So even though M2e is kind of that small protein on the surface, the antibodies we generate are still recognizing the whole virus. What's important is that we also get cellular responses, and so we have splenocyte proliferation. These spleen cells are producing interferon and IL-4 in the case of G2 and G3, but not in a serum or saline no control or the soluble protein. And so we're getting both cellular and antibody responses, which is really what you want for flu. And a lot of papers and a lot of research really only focuses on the antibody response, and for flu, the cellular response is really important as well. So we were excited to see this, that we were getting this balanced response, because with a lot of formulations, you really only get an antibody response, or they only measure an antibody response, which is hard to conclude what the cellular response is. And so this is really the thing that the initial response challenge that made us really excited about this platform and really kind of pushed forward the next generations. And so what I'm showing here on the top row is body weight of mice who have been immunized and then challenged with either the Philippines virus, which is identical to our peptide sequence, or on the right-hand side, the California virus, which has 4 amino acid mutations, which of that short sequence is the most divergent, really, that kind of exists. And so what you can see in both cases, the blue is soluble antigen, the green is no vaccine, and then the red and black are PNCs with or without the adjuvant. And so in the case of the Philippines, you see if you're vaccinated with soluble M2e, the animals lose weight quickly and they succumb to disease as if they were vaccinated with saline. So that soluble M2e protein really has no immunogenicity. Whereas the nanoparticles of both kinds are able to protect, the animals see a small weight drop against the identical M2e antigen, or virus containing the identical antigen. And if we go to the California side, which is the most divergent, interestingly, we see that our PNCs without the adjuvant actually protect all the animals, whereas with the adjuvant, we see death of the animals. And so that is, it's good, in the short term that our PNCs are protecting the animals, are not dying, but if you look at that weight loss, right, it's getting down to 20% or so. And so these animals are getting quite sick. So certainly this is better than dying, but it shows us that we have a lot of work to go in terms of protecting that response and ultimately preventing illness. And so if we moved on into generation 2, and I'll call these generations just to keep them straight, but it's not all necessarily working linearly because we're trying to study how the different antigens are playing a role here. And so the next thing that we look at is hemagglutinin. So I told you that hemagglutinin is one of the most immunogenic proteins on flu. And so we wanted to see if we could actually incorporate hemagglutinin into our vaccines, but using the stalk domain and not the widely mutating head domain. And so what we took is an HA domain from an H7 strain of flu, and we again in this case trimerized it. Hemagglutinin is a natural trimer. And so we need to be able to protect that structure when we make our particles. And so in this case, we made the particles with a reducible crosslinker, thinking that they could break up more easily. And we also gave an extra coating of hemagglutinin on the outside. As I kind of glossed over, but in that early data I showed you that there was different cytokine responses whether the particles were coated or not coated. And so one of the things that is not talked about a lot but is really important is that when you put nanomaterials in the body, proteins from the blood or the interstitial fluids or wherever it is, the lungs, proteins from the surrounding solution coat particles immediately. And so now you have something that is coated with the body's own proteins, which significantly affects the immune response. And so what we can do to kind of preempt that a little bit is make our own protein coating. And so by pre-coating with hemagglutinin, we know that we have now a surface of folded, properly folded hemagglutinin on the surface, and that may reduce the adsorption of other proteins from the body, which are not going to be helpful and may mask our particle. And so what we saw here, so just working with the hemagglutinin, on the top row, we investigated giving 1 or 2 doses, either intramuscular or intranasal, and we looked at just broadly neutralizing antibodies in panel A, and also hemagglutinin-specific inhibition in panel B. And what you can see is that certainly 2 doses is better than 1 dose, but also that intramuscular is better than intranasal. And so while we use intranasal for the first time, in the rest of the work that I'm going to show you, we're going to stick with intramuscular. But the good news at the bottom is that at least for most of the treatments, the 1 intranasal injection did not give 100% survival, but all of the others did, is that we were able to protect mice from challenge with the exact same strain. So an H7N9 strain challenge, and this was an H7 hemagglutinin. So in a perfect match. So this wasn't really asking about broad protection. This was asking, can we make an HA vaccine, as we're kind of stepping forward. Okay, so the other thing to note here, and it was great to bring up kind of this pharmacy and stability and things like that is really important. So proteins are notorious for being unstable, for aggregating, and having them sit around is not a good thing. And so what we wanted to do here is test whether or not we could have a formulation that would be stable, especially as these are going to be going all around the world, and we don't want to be able to be reliant on cold chain like you are for live and inactivated viruses. And so on the left, I just wanted to notice that other cytokines come up. So in this case, TNF was produced by the nanoparticles and not the soluble, whereas IL-1 beta is in other ones. And so, it really depends on the antigen and the exact response you see, but in all cases, you see differences. And so what I'm showing in the middle here is that if we store our particles at room temperature in saline on the bench for 3 months, we see basically no difference in the size distribution. And most importantly, when we look at the hemagglutinin inhibition titers of these animals, the aged, so 3 months aged, soluble on the benchtop, perform just as well as the fresh nanoparticles, all being better than soluble or PBS. And that dotted line, that's the World Health Organization kind of acceptable hemagglutinin inhibition titer level. So if you're not above that line, you're not a good flu response. So storage can be done. Okay, so the last two things are starting to combine multiple proteins. And so in this case, we took nucleoprotein, that internal flu protein, which is very conserved, with the M2e conserved protein, and mix these proteins together. And so you can either take the whole protein from each, or what we did is some kind of protein engineering. And so getting back to this idea that small peptides can give you a more specific responses, we concatenated several domains of nucleoprotein together, as well as M2e sequences from human and swine and bird. I can't read them all, but 4 different species. And so what we did is we first desolvated the nucleoprotein core, and then we put a coating of M2e around it. And so this is just what they look like. When we look at a challenge with, again, the same sort of the same strain of flu that these are coming from, although because it's conserved, the strain is less important here. You can see that the orange and the green are the nano formulations, either these concatenated peptides or the whole protein core and coated protect the animals in all cases, and the amount of illness is getting better. Whereas the soluble is the blue, and that one did not protect. Okay, so last one, I have 2 minutes left, is kind of where we are now, making this more complex in a spatial way. And so what we've done is we're trying now to take the benefits of HA and the benefits of these core conserved proteins and bring all of this together. And so the system that we have here is, again, this M2e fusion of multiple strains from humans, avian, pig, so on, and that's going to make the core. So we desolvate an M2e core. That's called the UNA-M or C. I'm just going to come over here. So here is the so this is the M2e fusion protein. We make a desolvated regular particle from that, cross-linked, and then we have 2 different hemagglutinin domains that we have fused together. So we have one from an H1 strain and one from an H3 strain, and we coat those on the outside. So this is now combining kind of those earlier generations where we have the M2 core and the HA coat. And the idea here is that we can have the M2E with all of these, and then we have either strain 1 HA, strain 3 HA, or the combination we write as 1, 3. And so this is the last slide of data, and what I just want to show is how we are now really getting protection against the cross coverage. And so we have the H1 data here, body weight and survival, and you can see that the animals that are surviving are the pink and the blue, the animals that have the HA and the M2E. So the yellow is only the M2E, so that alone is not enough to protect all animals. And if you have the HA1, either because you have it alone or with the third one, you're protected. The same case with the 3. The 3, either alone or in the 13, protects, while the M2E alone in this case is good enough to protect from an H3 and 2. Remember, this is the Philippines one, so this was the M2E sequence that matched. I think what's most exciting is over here is if we look now at an H5 strain or an H7 strain. So we have no HA from H5 or H7 strains in there, but we are now getting protection from these strains with the different formulations. And so what's interesting to notice here is that so H5 is more like H1 and H7 is more like H3. And so if you vaccinate with the HA1 or the combination, you get protection against both of these. And so by combining 2 forms of hemagglutinin, 4 strains of M2E, all in the same particle, we can get protection against a number of different strains. Okay, so I just like to conclude that you can make nanoparticles from different antigens and gain benefits in terms of the vaccine response and protection. Thank you.